Good evening. In this video, we're going to look at some recent research by Dr. B.J. Casey and her associates and related work by Dr. Ian Weaver. These research articles look at fear and how we react to fear. They combine to suggest an elegant weave of how brain architecture, genetics, epigenetics, history, and environment interact to form a plausible scaffolding for human experience. They also suggest how any of these factors can be a plausible pathogenic pathway in the development of mental illness.
Let's start off by defining some terms. Brain architecture refers to different structures inside the brain. The amygdala is a structure that we'll be talking about. It's an almond-shaped structure that's about 3/4 of an inch long. When I talk about environment, I'm talking about the elements outside of ourselves that we interact with. People, places. A baby's environment could be as small as a mother in one room. Your environment is probably a bit more complicated. Genetics is your genetic code. Epigenetics is how genes express themselves. The expression of many genes is impacted by the environment. When I was a kid, genetics determined your eye color and environment had nothing to do with it. It turns out that most of the genetic code is more complicated than that. Biologically sensitive periods are periods when we are biologically more receptive to certain types of learning. It is thought, for example, that it's much more difficult to master a second language after puberty, which is why we wait until middle school to teach second languages. In 2000, B.J. Casey, along with Jay Geed and Kathleen Thomas, published an article that was part of the beginning of the conversation on the neurological differences between adolescent and adult brains. For those of us working in juvenile justice, this conversation has shifted and continues to shape our profession. This was among the first articles to use fMRI imaging to show that the prefrontal cortex is the last part of the brain to mature. This research uses brain scans done on the same subjects over a period of 10 years to show how the brain matures. Researchers then compiled the images into movies to show the maturation process. Look at the area that I've got circled. That's the prefrontal cortex. Do you see how red that area is? Do you see how other areas are blue? The areas in blue are the most mature, the areas in red are the least developed. The 6-second movie that you're about to see shows 15 years of maturation in just 6 seconds. Are you ready for a little time travel? Here goes.
Prefrontal what? Let's develop a quick, easy model of the human brain using our hand. This model was first suggested by Daniel J. Siegel. Hold out your hand and tuck your thumb into the palm of your hand. Now wrap your fingers around your thumb. What you wind up with is a pretty serviceable model of the human brain. Imagine that your arm is your spinal cord and that the fleshy part of your hand down here is the brainstem, and that the brainstem handles most of the automatic stuff: breathing, heart rate, digestion, sleep, all the stuff that happens largely beneath consciousness that you don't need to think about all the time, but it just keeps working anyway. Now, in this model, your thumb is the limbic system. And the limbic system is where emotions and memory start. We're particularly interested in one part of the limbic system called the amygdala. You can think of the amygdala as your inner Alfred Hitchcock. That's where fear starts. The area surrounding the limbic system is your cerebral cortex. And the cerebral cortex is that area which is developed most in mammals and most in humans. It gives us the ability to remember and make 3D representations of the world that we use in planning, imagination, moral reasoning. We're particularly interested in one part of the cerebral cortex called the ventromedial prefrontal cortex. And we'll get back to that in a minute. But right now, I want you to consider the location of the limbic system and how it's close to the brainstem, but also it's close to any part of the cerebral cortex. Now remember that the limbic system is where emotions reside. So when emotions are involved, they get in the middle of everything.
The part of the cerebral cortex that we're concerned about here is the ventromedial prefrontal cortex. Let's say that that's represented by the fingernails in your ring and middle fingers. Although the prefrontal cortex is involved in a great many aspects of behavior that are fundamental to our experience, these articles are looking specifically at the part the prefrontal cortex plays in fear modulation. Here we can already see two elements of the weave that Dr. Casey and her colleagues elucidate in these articles. The first element is that the interaction between brain structures forms the architecture of the brain that creates conscious experience. The second is that different parts of the brain mature at different rates. Treating the Developing Brain, published in 2013, returns to the differences in maturations of the amygdala, the Hitchcock bit, and the prefrontal cortex, the reasoning bit. The article has a graphic that should be very close to any juvenile justice professional's heart. The top line represents the maturation of the emotional part of the brain. The bottom line represents the maturation in the prefrontal cortex that's responsible for reason. Note that the prefrontal cortex is not fully developed during adolescence. The difference between these two maturation rates is the area in yellow. The area in yellow is the biological reason people working in juvenile justice have jobs.
Let's digress on that for just a moment. Obviously, there are periods during our lives when we're better at learning things than we are during other periods. Cue up any manner of cliche, including babies have to learn to crawl before they can walk. But it turns out that there are biologically critical periods where, if a structure in an organism isn't exposed to the proper external stimuli, that structure never develops properly. The classic example of this is research done back in 1963 by David Hugel and Torsten Wiesel. They sewed shut one eye of a newborn kitten and left it sewed shut for 3 months. When they removed the sutures, they noticed that the kitten could not see through the eye that had been sewn shut. Furthermore, even though there was nothing structurally wrong with the eye, the kittens never regained eyesight in that eye. So, if the eye is deprived of light during that critical 3-month period, eyesight never develops. This is also an example of research done back in the 60s, which in retrospect is downright creepy.
Researchers in Wales recently used the same method to study lazy eye and got bad press in the process.
I have a nightmare about human development that loops back to the whole concept of critical biological periods. Is empathy in humans hardwired, or is it dependent upon some biologically critical period during which, if an infant doesn't get the nutrition and physical touch and sound stimulation that are part and parcel of a mother's love, will that infant never develop empathy? The Dalai Lama says that we're all born with the seeds of basic goodness. But what if those seeds aren't watered? The question haunts me.
There is some fascinating work related to this question. In 2004, a research team led by Dr. Ian Weaver published a study that demonstrated that maternal behavior during the first 6 biologically critical days of a rat pup's life impacts the expression of genes that alter the hypothalamic pituitary adrenal response to stressful events. Pups who were raised by dames who spent more time nursing and grooming their pups, nurturing dames, raised pups who were behaviorally less fearful under conditions of stress when they became adult rats, compared with pups who were raised by dames who spent little time nursing and grooming their pups, anxious dames. But what if the pups were switched at birth? What if we took the pups born of an anxious dame and gave them to a nurturing dame to raise during that first 6 critical day window? The pups would grow up to be adults who exhibited less fear under stressful conditions. And what would happen if we placed the pups from the nurturing dame and had them raised by an anxious dame? They would grow up to be more fearful under stressful conditions.
Clearly, licking and grooming behavior during that critical first 6 days of life has an impact on the stress response in adult rats. But biologically, how does that happen, and how is it maintained? 2 years after publishing the article we just discussed, Weaver and his associates published the article Epigenetic Programming and Maternal Behavior and Pharmacological Intervention, Nature versus Nurture. Let's call the whole thing off. This is what happens with the rats.
One gene in rats has an impact on how sensitive cells in the hippocampus are to stress hormones. Think of this gene as having an on-off switch. 2 to 4 days before birth, these genes are set in the on position. At birth, the switch is automatically turned off. The switch is turned back to the on position in pups with nurturing dames by the licking and grooming behavior. The switch is not turned on in pups raised by anxious dames because the dames don't engage in enough licking and grooming behavior to switch the gene back on. So it isn't nature versus nurture, it's the combination of nature and nurture. This research has all the elements of the model I am proposing. One structure in the rat brain, the hypothalamus, is biologically sensitive during the first 6 days of life to environmental conditions, licking and grooming behavior by the dame, which impacts whether the certain gene is changed from the off to the on position, epigenetics, which has a lasting impact on how fearful the adult rat will behave under stressful conditions.
It's not that this research answers the question that haunts me, but this research does demonstrate that in mammals, nurturing behavior during the first critical days after birth impacts how a gene is expressed. And that leads us away from the polemics nature versus nurture arguments towards something that could be measured. And when science develops technologies to measure a problem, progress becomes possible. Now, granted, this is just one gene in one mammal, but it does show that the default setting for rats is to become adults who are fearful under stressful conditions. And that rats who have nurturing mothers are less fearful under stressful conditions because of the behavior of the mother rat. Now, painting with a very broad brush, this is like saying that rats are born with the seeds of basic goodness. But those seeds have to be nurtured in order to manifest as adult behavior. Okay, that's fine about rats. But what about humans? This takes us back to the articles by B.J. Casey and her associates. These articles look at mammalian brain circuitry for fear and how that circuitry is differentially sensitive during different periods of psychosocial development. These articles look at fear circuitry during childhood, adolescence, and adulthood. Fear circuitry in mammals is highly conserved, which is to say, the circuits that fire off when a mouse feels fear are the same circuits that fire off when a human reports feeling fear. If you think about it from an evolutionary standpoint, this makes a lot of sense. Adaptive fear is critical to survival. A mutant organism that didn't feel fear might have a pleasant life, but it probably wouldn't live long enough to pass along its genes. Because the fear circuits in both mice and humans are so similar, this research uses experiments on both mice and humans to develop a better understanding of how fear learning works. The use of animal experiments to improve our understanding of human psychology is called translational research. By examining fear on the genetic, neurological, and behavioral level in both mice and humans, B.J. Casey and her associates have developed a more detailed and more robust understanding of fear learning than would otherwise be possible.
The process of fear learning and unlearning is familiar. Let's posit a boy who does not exhibit a fear of dogs until he is bitten. The boy then generalizes the fear to all dogs in situations where no threat exists. Exposure therapy is utilized to help the boy differentiate between situations where the dog is dangerous from situations where fear is not warranted. Initially, when the boy encounters a dog, the dog does not bite. The reaction is neutral. Then the boy encounters a vicious dog who bites the boy. Result: fear.
Did your heart rate jump just now? That was your inner Alfred.
The boy encounters the first dog and, although the first dog does not bite, the boy is terrified. This is, of course, traumatic fear learning. The boy is terrified of all dogs regardless of whether the dog poses a danger or not. Under careful supervision, the boy is then exposed to the non-biting dog for short periods while the dog is being held on a leash. Result: less fear. Another exposure to a slightly longer period. Result: substantially less fear. As the youth tolerates increasingly longer exposures to fear, the level of fear reported by the boy continues to decrease, until eventually, as he practices correctly identifying pictures of dog exhibiting aggressive behavior, is coupled with sessions and being in the same room with a safe dog, the child has a neutral reaction to dogs who are not aggressive.
You'll often hear this referred to in the literature as extinguishing the fear response. I'd like to draw a distinction here. We've taught the boy a new behavior of not feeling fear when there's no danger present. That's different from teaching the boy not to feel fear. When faced with a dog who's showing aggressive behavior, we've reinforced the idea that feeling fear is appropriate. Not feeling fear automatically when there's no danger present is a second, separate skill set. It doesn't preclude fear. It simply provides a new response for figuring out when fear is appropriate and when it's not. Only exposure therapy doesn't work for a lot of people. The research in this review suggests that differences in brain circuits contributes to our inability to learn when fear is no longer needed. It also shows that differences in brain circuitry are more pronounced during adolescence than during childhood or adulthood. How changes in one specific gene can make it more difficult for some people to unlearn fear than others, and how childhood traumatic experiences can impact how that gene functions. Let's look at the problem from the bottom up. We'll start with genetics and build our way out to how psychosocial functioning is impacted over time. One aspect of fear learning that this research explores is the relationship between BDNF and fear learning. Brain-derived neurotrophic factor, BDNF, is a protein that plays a critical role in neuronal health, how neurons grow, and how sensitive the connections between neurons are to changing conditions. Now, the ability to adapt to changing conditions is a pretty good definition of learning itself, so we're dealing with a fundamental process here. And perhaps because it is a fundamental process, BDNF is often not facetiously referred to as the miracle grow of the brain. One gene impacts how much BDNF is available to your brain. Scientists have come up with a designer version of this gene in mice that allows them to directly test the impact of stress on mice development over time, and to test it in ways that we couldn't possibly do in humans.
So what could they do with mice that they wouldn't do with humans? After they stressed them, they'd kill them. They'd pop out their brains. They'd slice up the brains and then they'd look at them under a microscope. Humans don't like that.
The genetic combination that results in the most BDNF being available to your brain is the valine nucleotide, often shortened to VAL. And the genetic combination that makes the least amount of BDNF available to your brain is the methionine nucleotide, often shortened to MET. Now, I need you to access your junior high school biology brain. Remember, that genes come in pairs, that one comes from your mother and one comes from your father. So it's possible to have a Val-Val combination if both your mother and your father contributed the valine nucleotide. It would be possible to have a Val-Met combination, a Met-Val combination, or a Met-Met combination. The Val-Val combination is the one that makes the most BDNF available to your brain.
Theoretically, we would expect to have 25% of the population to have two Val alleles, 25% to have one Met and one Val allele, 25% to have one Val and one Met allele, and 25% to have two Met alleles. We can simplify this by combining the combinations of one Met and one Val allele to demonstrate that 25% of the population has all Met alleles, 50% has a combination of Met and Val alleles, and 25% would have all Val alleles. But it rarely works out like that. An article published in 2008 in the Croatian Medical Journal showed that while the Met-Val combination occurs in 46% of Korean population sampled, which lines up with the 50% that we would expect in a random distribution, only 32% of the Croatian population had a Met-Val combination. In the Croatian population, the Met-Met combination was rare, while the Val-Val combination was the most common.
Genetic combinations that involve the Met allele have been associated with impairments in certain types of memory formation and learning, and these impairments are associated with increased risk of depression, anxiety disorders, and PTSD. In research published in 2010, Solomon et al. Did equivalent research with mice and with humans that demonstrated the relationship between the Met-Val polymorphism and fear extinction. Genetically engineered mice were used that mimic the Val-Met polymorphism in humans. The mice were played a tone for 30 seconds with an electrical shock during the last second of the tone administration. This was done for 3 times, and then the mice were given a 3-minute rest, 4-minute rest, and then they heard the tone for a 30-second period with no shock, 30 seconds of rest, and then another 30-second administration with no shock, and the no shock condition was done for 30 times. The mice were filmed during the experiment, and the films were reviewed for the percentage time that mice spent having a freezing posture. Freezing posture is when they're crouched and there's no movement. This is the freeze part of the fight-flight-freeze response. Now, mice with the genetically engineered Met-Met combination spend a significantly larger amount of time in the freeze posture than mice with the genetically engineered Val-Val combination. Simply put, once the Met-Met mice were shocked and heard the tone, they stayed scared whenever they heard the tone, whether there was a shock or not. The Met-Met gene mice were less good at learning to adapt to new situations than the Val-Val mice.
When they did the equivalent experiment with humans, do you think they used electrical shocks? No, I don't think so.
Nope, they didn't shock the humans. Instead, they showed the humans colored squares, either yellow or blue, for a 30-second period, and during the last second of each administration, they played a loud tone, a tone at 1,000 hertz at 95 decibels. Now, 95 decibels is about as loud as a jackhammer sounds from 50 feet away. So it's a loud, even slightly painful sound, but it didn't hurt anybody's hearing. And instead of measuring crouching behavior during these tests, they continuously measured skin conductance. But the impact of the Val-Met polymorphism varies across time. BDNF levels are lowest during childhood, and then rise by about 1/3 during late teens, and then remain fairly level. The combination of genetic risk and rising BDNF levels combine to form a dynamic risk over the lifespan. A child whose BDNF levels are already low is more likely to sustain severe nerve damage from a traumatic event such as abuse and neglect If that child is a MET carrier instead of a VAL carrier, the VAL carriers provide more support for damaged nerves. In adolescence, it could be that the MET allele is actually a protective factor. Adolescence is a time of risk-taking. Adolescents with the MET allele are more reactive to stress and may actually avoid risky situations more than youth with the VAL allele. So the same genetic combination that forms a risk during childhood could also be a protective factor during adolescence. When it comes to fear learning and fear unlearning, it appears that adolescence is a period of impaired learning.
Hey, are you in there? I just said that teens don't appear to learn from experience. You're really surprised about that, aren't you?
In a follow-up to the 2009 research, Treating the Developing Brain, published in 2013, looks at fear learning in children, adolescents, and adults. Like the previous research, this research pairs colored squares with an aversive sound. Adolescents continue to react to the colored squares without the aversive sound for a much longer period of time than either children or adults. Children and adults were more able to adapt to new situations than adolescents were. It appears that adolescence is a biologically sensitive period where adaptive learning is impaired. In that this is a description of how exposure therapy works, it raises questions about the efficacy of exposure therapy with adolescents.
In a related article, Drysdale et al. Review the data from the Child-Adolescent Anxiety Multimodal Study to compare how effective cognitive behavior therapy was compared to placebo in children, adolescents, and adults. The results demonstrated a similar pattern of CBT being less effective with adolescents than with children or adults. Drysdale et al. Point out a couple of big caveats with this research. The Child-Adolescent Anxiety Multimodal Study wasn't designed to show differences in treatment efficacy of CBT between age groups. CBT was one component in a larger treatment protocol in the multimodal study. The effects of CBT can't be separated out. The multimodal study also had fewer adolescents than children, which reduces the statistical power of the test, which leads to the main caveat. The differences between adolescents and children were not statistically significant, but they are suggestive.
Returning to treating the developing brain, Casey used adolescent mice in this research. She showed that adolescent mice have the same deficit in fear extinction learning as human adolescents. But here's where the benefit of translational research begins to pay off. She cites research by McCallum in 2010 that showed an equivalent problem with adolescent rats. McCallum showed a drop in synaptic density in the prefrontal cortex in both humans and rats that caused a naturally occurring lesion during adolescence. There was a drop of perhaps 50% in the synaptic density during adolescence. Can't you just hear Bill Cosby saying something about brain-damaged adolescents?
But the reason why the curse works is because all children have brain damage.
Well, he may not have been kidding. But McCallum goes on to show that an injection of D-cycloserine directly into the prefrontal cortex of the adolescent rats improved fear extinction learning to a level equivalent of adult rats. Now, can you just see us putting an injection of D-cycloserine directly into the forehead of humans? You see, that's why translational research is valuable. We can do that with rats.
It turns out that D-cycloserine, under the brand name Ceramycin, has been used for years as a second-line medication for treating TB. It has also been shown to enhance traditional therapies for acrophobia, fear of heights, and obsessive-compulsive disorder. While these findings are encouraging, it is probably the case that memory and complex trauma is not the same as with specific behavior phobias like OCB or acrophobia, or even fear of dogs for that matter. In research published in March of 2014, Sharinga et al. Explored the use of D-cycloserine to augment traditional cognitive behavioral therapy in PTSD in 7 to 17-year-old youth. Authors warn, quote, "It appears that augmentation with DSC represents unique challenges in PTSD. Because PTSD involves complex, life-threatening trauma memories, as opposed to imagined dreadful outcomes of other anxiety disorders, the use of DSC may require greater attention to how it's used, coupled with exposure-based techniques. DSC may have inadvertently enhanced reconsolidation of trauma memories rather than more positive and adaptive memories."
So, while this research suggests that the cause of decline in synaptic density in the prefrontal cortex during adolescence is amendable by pharmacological treatment, it probably isn't going to be as simple as cranking up the seramycin. The problem with the D-cycloserine studies is that the medication was administered systemically into the bloodstream. So we can't really figure out which parts of the brain were impacted more. Remember that we started this essay by showing a graph that showed that the amygdala matures at an earlier age than the prefrontal cortex. The amygdala is the top line, the prefrontal cortex is the bottom line. Remember that the amygdala is your inner Alfred Hitchcock. It generates fear. Well, the prefrontal cortex is capable of suppressing that fear when the context is inappropriate. The connections between the amygdala and the prefrontal cortex have a lot to do with how well we can unlearn fear when fear is no longer appropriate. Fear extinction. The health of that circuitry is absolutely critical. The hippocampus also plays a part in this process. Remember that the hippocampus is part of the limbic system, your thumb in this model.
Let's turn to the architecture of fear. In these diagrams, VMPFC stands for the ventromedial prefrontal cortex. This is a simple-minded wiring diagram of fear signals in the nervous system. When the amygdala determines that an environmental input is dangerous, signals from the amygdala trigger the physiological expression of fear. Research by Joseph Ledoux, famously cited by Daniel Goleman in Emotional Intelligence, demonstrated that the amygdala, our inner Alfred, has a privileged status in the brain. When environmental input is perceived as dangerous by the amygdala, it can send fear signals to the rest of the body before the ventromedial prefrontal cortex or the hippocampus can even become involved. As the amygdala sends fear signals to the rest of the body, it sends signals to the ventromedial prefrontal cortex and to the hippocampus, which begin the process of building memories of what is dangerous about the situation so that the brain can do a better job of anticipating danger the next time you find yourself in a similar situation. In this instance, doing a better job of anticipating danger means experiencing fear more intensely for a more sustained period of time. This learning to fear a situation that has become dangerous in the past occurs because signals from the ventromedial prefrontal cortex and the hippocampus are available during subsequent episodes. The coordinated information between the three brain structures makes for a strong, robust fear response. The circuitry between the amygdala and the frontomedial prefrontal cortex form a brake accelerator mechanism for fear. Neuronal signals from the amygdala and the prefrontal cortex can have the impact of reinforcing fear. Signals from the amygdala travel to a part of the prefrontal cortex that sends a reciprocal signal back to the amygdala, which amplifies the fear response. But the prefrontal cortex, after evaluating the risk, can also send a reciprocal signal back to the amygdala that decreases the fear signal. So the prefrontal cortex has the ability to, in conjunction with information from the hippocampus, evaluate the signal from the amygdala and decide to either stomp on the gas or put on the brakes. But only if the circuitry between the amygdala and the prefrontal cortex is working properly. Let's look at the child who was bitten by the vicious dog sequence in terms of brain architecture. Child is bitten by the dog, and the boy develops a fear of dogs. The visual signal of a dog immediately fires up the amygdala, which simultaneously sets off a bodily sense of fear but also sends a signal to the ventromedial prefrontal cortex so that the memory is available for future reference. Now let's say the boy encounters another dog, but the dog doesn't bite. The environmental signal of a dog who isn't biting shoots to the amygdala. The amygdala then creates a bodily sense of fear, but also sends a signal to the prefrontal cortex, which now has information that dogs don't always bite. Given another encounter with a dog that doesn't bite, the hippocampus now has information about dogs that bite and dogs that don't bite, and the hippocampus sends the information both to the amygdala and the prefrontal cortex. The amygdala sends a danger signal to the prefrontal cortex, which evaluates the difference between this signal and the time when the child was bit. The prefrontal cortex then sends a signal back to the amygdala that has the effect of lessening the fear response. Subsequent encounters with the dog that don't bite, without any instances of a dog that does bite, strengthen the dampening signal from the prefrontal cortex, and the fear response continues to diminish. This is fear unlearning, or the fear extinction process. But what happens when the circuitry between the amygdala and the prefrontal cortex is impaired? The signal may go up to the prefrontal cortex, but the brakes never go on. So even though subsequent exposures to dogs subsequently results in not being bit, the fear response just keeps getting stronger. This is the fear unlearning part not working.
These patients were more likely to experience unremitting fear even in the absence of a current danger. Fear without danger is a pretty good definition of anxiety. Hartley and Casey in 2013 demonstrated that the integrity of the fibers between the amygdala and the ventromedial part of the prefrontal cortex are inversely correlated with trait anxiety. In other words, people who have poor connections between the amygdala and the ventromedial part of the prefrontal cortex are more likely to view the world through an anxious lens. Solomon in 2010 showed the intersection between brain architecture and genetics. The fMRI studies in that research showed that subjects who had the meta-allele, that's the version that allows less BDNF to be available to the brain, those subjects showed less activation in the prefrontal cortex and more activation in the amygdala than subjects with the valine version, the version that makes more BDNF available. Subjects with the meta-allele showed significantly slower fear extinction. That puts us in a position to return to my original question. What are the neurological impacts of neglect on childhood development? While this research doesn't provide us with as conclusive evidence as, say, Weaver's does, it does provide some intriguing evidence. Now, Weaver was able to provide conclusive evidence on the impact of maternal behavior on rat pups because he could scientifically control the amount of stress that the rats were exposed to, and he could control the amount of time that they were exposed. After that amount of time, he could then autopsy the brains. That raises two problems with humans. First of all, we can't scientifically control the amount of stress that infants are exposed to. That would be wrong. And secondly, thankfully, there just aren't that many opportunities to autopsy human infants. While our technology to look inside the human brain has dramatically improved over the last 20 years, we still can't get as fine a look into what's happening in the human brain as we can with an autopsy. Perhaps in the near future, that technology will develop. But by looking at the experience of infants raised in Romanian orphanages, we are able to take a look at the interaction between environment and genetics and see the outcomes as never before.
Under the Ceausescu regime in Romania, both abortion and contraception were banned. Many children were abandoned to orphanages where the conditions were beyond wretched. Aid workers arriving in the orphanages after the fall of Ceausescu found youth housed 3 or 4 to a bed. There were no medicines or washing facilities, and physical, emotional, and sexual abuse were common. Neglect was the standard of care. One group of children were found in a basement where they had not seen natural light for years. The children in these pictures look like they're from Dachau in World War II, but they aren't. They're from Romania in 1990.
The histories of these youth creates a naturally occurring science experiment. Youth who were raised in Romanian orphanages were compared with a group of children from the United States who were not raised in orphanages. fMRI brain scans were taken of all the youth, and comparisons were made between the youth who were raised in an orphanage and the youth who were not raised in an orphanage. There was no difference between the two groups. But, you knew there was a but, if the Val-Met polymorphism is taken into account, there was a difference. Youth with the Met allele, that's the allele that produces less BDNF, had smaller brain volumes than youth with the Val allele. In other words, youth with the Met allele were less able to deal with the toxic stress of Romanian orphanages than were the other youth. But wait, there's more. The data in this study was linked with the data that we talked about earlier about differences in brain structures. The Romanian orphans were subdivided into two groups. Those orphans who were adopted at 15 months of age or earlier, called the early adopters, and youth who were adopted at more than 15 months of age, the late adopters. The late adopters were subject to a greater level of stress than the early adopters. Now, when they compared brain volumes, the brain volumes were the same. But when they compared the amygdala of the different groups, there was a difference. The amygdala in the late adopters, those who were exposed to stress longer, was larger than the amygdala in the early adopters. And the amygdala in the early adopters was larger than the control group. For those of you who don't remember, the amygdala is your inner Alfred Hitchcock. These studies showed that children with heightened activity in their inner Alfred was associated with poorer eye contact between children and their parents. The orphan children were also slower to distinguish between sad, fearful, happy, and angry faces. Orphan children made more errors in recognizing sad, fearful, or angry faces. The orphan group and the control group were equally skilled at recognizing happy faces.
This research shows how 5 factors combine to influence how well children and adolescents are able to adapt to a situation that was once dangerous but is no longer a cause for concern. Consider how this ability to adapt to changing circumstances can be considered as an exemplar of healthy human mental functioning, and all the ways that this functioning can become impaired. Neurological development takes place within the context of time. The orange portion of the graphic represents a biologically sensitive period around 15 years of age. These articles have shown how genetic variations impact the availability of BDNF, or Miracle-Gro, within the brain. This genetic polymorphism can impact brain architecture. Remember how the presence of the Met allele is related to less extinction learning and less activity in the prefrontal cortex and more activity in the amygdala? This is an example of genetics impacting brain architecture. An individual with the Met allele gene who had been exposed to high levels of trauma might well have difficulty learning to feel safe in situations that were not threatening. This person is likely to have a "life is dangerous" button stuck in the on position.
By definition, epigenetics is the combined impact of environment and genetics. The adoption study suggested that the presence of the Met allele only made a difference in youth when they were exposed to trauma for an extended period of time. Adopted youth with the Met allele who were adopted at an early age didn't show significant behavior differences with youth who were not adopted with the Met allele. But youth who were adopted at a later age and had the Met allele did have significant behavior problems compared to youth who had the Met allele who weren't adopted. That's a pretty long string of ideas. It might be simpler if we can visualize what's happening. Let's imagine that these cups represent the amount of stress that a brain can absorb without overflowing and causing possible nerve damage. Let's imagine that the bigger cup represents the vowel carriers, and the vowel carriers have more BDNF available than the met carriers, who have relatively less BDNF available and therefore can absorb less stress. Let's say that this lovely stuff that was red originally represents a normal episode of stress. The vowel carrier can easily absorb that much stress without overflowing. The met carrier is full, but doesn't overflow. So, a normal amount of stress can be easily absorbed by either genetic combination. It doesn't matter. Either combination is good enough. But, imagine that we have a single, very stressful episode. And this is a single, very stressful episode. The vowel carrier can still absorb it. No overflow. No toxic mess. But the met carrier can absorb it all, and it begins to overflow. And the more overflow, the higher the likelihood for nerve damage. But what would happen to our little cup subjects if they were subjected to early chronic childhood stress? During childhood, the BDNF levels are already lower, and we know that chronic stress causes the gene to express less. So whether you're a vowel carrier or a met carrier, you're going to have relatively less BDNF available during that time when there's naturally not as much to begin with. So a vowel carrier who normally might be able to absorb this much stress, under the epigenetic condition of chronic stress, might only be able to absorb this much. And the met carrier, who could only absorb this much under the best of conditions, now can only absorb this much. Now, if we subjected both subjects to a normal dose of stress, the vowel carrier doesn't overflow. There's still no toxic mix. But for the met carrier, even the normal amount of stress causes an overflow. Now, any subsequent episodes of stress, and we're saying this is chronically stressful, so they're happening often, would cause the vowel carrier to overflow. But every single episode that the met carrier is exposed to causes a little more toxic mess to build up. And eventually, or maybe not eventually, it's going to result in nerve damage.
So genetics impacts brain structure. The combination of genetics and environment combine to determine the extent to which genes are turned fully on or only partially on. Epigenetics.
All of these processes impact brain architecture, and they impact brain architecture before consciousness. Now, I'm not trying to suggest that direct experience isn't possible. If I'm preparing food and I cut myself with a knife, the environmental impact on my body is direct and immediate. I don't get cut because I perceive myself being cut. I get cut because the knife is sharp.
But here we're more concerned with perception and want to note the elaborate interaction between environment, genetics, and brain architecture that occurs before consciousness even happens. When we put the interactions of the four factors that impact conscious experience together, we can see that there's a lot going on. And what is represented here is an idiot's simplification. We've looked at some of the implications of one gene variation that affects the availability of one molecule that impacts nerve health. This is the epigenetic brain architecture connection of the model. But the impact of BDNF differs among different brain structures. BDNF seems to have a particularly large impact on the hippocampus, most heavily involved in memory formation, and on the amygdala. Furthermore, BDNF is one of thousands of genetically influenced chemicals in the brain. The impact of one gene is small. It is the way these agents work in concert that has such a dramatic impact on how we consciously experience life. All of these interactions happen within the context of human development. In addition to the impact that the interaction between genes and environment have on BDNF availability, BDNF itself varies across the lifespan. BDNF levels are relatively low for infants and in childhood, peak in young adulthood, and then remain fairly level. So if we combine the already lower levels of BDNF in early childhood with the lower availability caused by the meta-allele, we can see how early childhood is a biologically sensitive period to chronic stress when compared to adolescence, when BDNF levels are higher to begin with. Add on to this what could be a risk factor for one mental disorder could also be a protective factor in a different mental disorder during adolescence. The same meta-allele that has such a pathogenic impact on the brain of children could be a protective factor in regard to substance abuse during adolescence. So let's get all these factors moving together at once across time. That's a lot of arrows flying around. Gee, there's so much going on here, this could be a schematic for just about anything. This could be the graphic representation of a deranged muskrat on steroids learning disco. But if any part of our model is defective, our muskrat could wind up doing a faceplant.
But there are so many different ways that a muskrat can faceplant. Some faceplants are fast, some happen in slow motion. In this video, we've discussed how brain architecture, genetics, environment, epigenetics, and biologically sensitive periods can all be pathological pathways for the development of mental illness. That's a lot of different combinations. 5 factorial, that's 120 different ways that those 5 factors could combine. This is a different way of understanding the relationship between genetics and environment and behavior than our current classification system allows for. Sometimes our current classification system works fine. A single traumatic event can lead to changes in behavior that would meet the diagnostic criteria for post-traumatic stress disorder. And that would be an example of a fast faceplant. But more typically, changes early in life lead to full-blown diagnoses that don't happen until young adulthood. An 8-year-old who suffers a series of traumatic events shifts from having an average disposition to having an anxious disposition. And that puts down the neurological groundwork for a major depressive episode that doesn't happen until the kid hits 30. That's an example of the slow, insidious faceplant that our current system doesn't have a way of recognizing. Our current system also doesn't do a very good job of conceptualizing mental illness. It tends to be pretty linear. As in the example I just gave you, trauma leads to changes in disposition that leads to depression. But the same trauma that led to depression could just as well be a prodromal event to the development of obsessive-compulsive behaviors, or phobias, or psychosis. This model has obvious treatment implications. The same 5 factors that are pathogenic pathways to the development of mental illness are also avenues for treatment. Pharmacological treatment has tremendous credibility in our society and is very convenient. Pop a pill in your mouth and you'll feel better. But what if the primary drivers of poor psychosocial functioning are environmental? Imagine a youth who is clinically depressed and lives in a neglectful environment. Does it really make sense to simply give that youth an antidepressant? Why would that resolve the situation? Living in a neglectful environment is depressing. A combination of individual therapy and family therapy would make a lot more sense. And considering the vast continuum of interactions that can occur between these 5 factors, does it really make sense to think about mental illness as something that happens in discrete diagnostic categories? Imagine a guy who doesn't quite meet all the criteria for a major depressive disorder, so we withhold treatment. That makes about as much sense as not treating someone for cardiovascular disease until they have a heart attack. We need to develop better diagnostic tools that allow us to identify impaired neurobiological functioning in much the same way as we have screening instruments for cardiovascular disease. But we also just need to flip the argument. We need to concentrate on conditions that help societies thrive. We know that stronger families prevent mental illness, that safe neighborhoods prevent mental illness, that lower air pollution prevents mental illness, that strong, sustainable economies, equitable economies, prevent mental illness. Programs that train youth in social and emotional intelligence, in addition to traditional IQ, prevent mental illness. All of the factors that we've identified as pathogenic avenues to mental illness can also be pathways to building stronger societies. So let's stop waiting for people to have emotional heart attacks. Let's get on with the business of building societies that thrive.